thank you for the invitation. I'm happy to tell you guys that we're a select audience here, so do feel free to interrupt and ask questions along the way. So my interest has been in really solving this problem, which is, this is a picture of a Drosophila embryo. The blue stains the actin in the muscles, and the red is staining the integrin adhesion sites, which you'll hear a lot more about. But really, what's amazing is the pattern of muscles in the developing embryo. And I'm just kind of fascinated by how the DNA manages to encode the instructions to make an embryo and make the cells of the right size and the right shape. And the way I've kind of approached tackling that is to focus on one bit of the machinery, which is the cell adhesion machinery, and see how that contributes to that process and try and understand that machinery. Our interest really is in cell adhesion, and we've got cell adhesion mediated by a class of molecules called cadherins, and then adhesion to the extracellular matrix mediated by a class of molecules called integrins. Actually, move my handy laser pointer out of the way. And so we've been sort of interested in how these interact with the cytoskeletal elements, drawn crudely here, the microtubules and the actin cytoskeleton. And really, we've pushed on using the combination of molecular genetics and imaging to tackle these problems. Integrins are much like any transmembrane receptor. They receive signals from outside. Do we have to have this part?
Oh, yeah. Yes, I know.
It's just a little. Right. Okay.
There we go. Yeah.
It was interesting when I was lecturing on cell fate decisions, but it was translated as the problem of self-hate, which was much more complicated. Okay, so they receive signals from outside, transmit information inside. The signal is a bit more complicated because rather than being a small secreted protein, for example, it's a big glom of extracellular matrix, big secreted proteins stuck to each other. And the response is largely organizing the cell cytoskeleton in the cell, although it can also transmit information to the cytoskeleton. I'm going to turn that one off too. You all know you're being recorded. But what I was finding fascinating about this particular class of receptors is that the information works the other way. So the cell can position where the integrins are, and the integrins help to organize the extracellular matrix, so they're organizing their ligand. So it's an interesting crosstalk between inside and outside to assemble these adhesion structures. So while you're listening, integrins are doing important things for you. So this is one of the reasons you should care about them. So they're sticking your epidermis down to the dermis. They're helping your leukocytes fight off infections and poised to help your platelets clot your blood. We've been using genetics to really discover the roles of integrins in development and to try and understand the machinery, particularly these intracellular, what we call IEPs, integrin-associated proteins that work with integrins to mediate their mechanism of action. And really my two big take-home lessons of this talk is that we discovered that the machinery works differently in different contexts. So if you look at different developmental events, the way it works is different. And then also if we start bringing in evolution, it helps us to understand how a very complex machinery was built up gradually during evolution and invented. And that helps us understand a bit what the different components contribute. So if you're not already a geneticist, just to highlight, the key thing about genetics is it allows you to compare two individuals. Your normal individual, for example, a normal fruit fly has about 15,000 genes, and a different individual which has one missing. And the difference between those two individuals is the mutant phenotype, so it may look different or the physiology might be different. And that allows you to infer what the function of the missing products are. So, for example, coming back to this slide, the reason why I can say this with confidence is because there are human hereditary diseases caused by mutations in the different integrin genes that cause either epidermal bullosis, very severe skin blistering disease, leukocyte adhesion deficiency, none of the leukocytes fight off bacterial infections, and Glanzmann's thrombocythemia is a very severe hemophilia. But also integrins are required earlier in development, and if you completely knock out integrins, you wouldn't get you or me, a person. So we've been using flies really to explore these kind of embryonic functions. And so here is normal development on this side, and this is from embryos that lack integrins. And I've kind of highlighted 3 different developmental events. So in this one, these 2 streams of cells are in the process of migrating toward each other to reform an epithelia that will eventually form the gut. And you can see in the absence of integrins, they're not moving and they fail to make that transition. This is the epidermal surface of the embryo, which normally is sealed up nicely along the dorsal midline, but that fails in an integrin mutant and it rips open. So it's required for the morphogenesis of the embryo. And then also for tissue assembly here, and what I've highlighted is the muscles attaching to extracellular matrix at the ends of the muscles. And if that's defective, then once they start contracting, they round up. So just to show you a little bit what the embryos look like live, this is an embryo. At this stage, the muscles are really just forming. They're labeled with the actin RFP. And then we've got talin GFP, which you'll hear more about shortly, which is one of these intracellular proteins that associates with integrins. Basically what's happening is just we're watching it develop, and you can see these attachment sites form, and then the muscles start to be contractile and pull on this more and more. And obviously, if you don't have those attachments there, then the muscles detach and round up. Okay, so another model that we found very useful from a genetic perspective is the formation of the wing. So you may not know a fly wing arises from a single flat sheet of epithelial cells that during metamorphosis in the pupa kind of everts like pushing out a sock. It makes a flattened sac, and then basically the basal layers of the dorsal and ventral surfaces come together and stick together. And if you look at a pupal wing and stain for integrins, you see these spots, which are the points where the dorsal cells are attaching via integrins to the ventral cells through an extracellular matrix. And these gaps are where the future veins form in the wing. And then in this case, what we've done is to use a genetic trick to just make a patch of cells in the wing mutant for integrin, because as I showed you, if you completely remove integrins, they die as embryos. But with that, you can see that you get a wing blister phenotype. I think we better not update this now. Right. So again, coming back to these sort of muscle attachment sites, the kind of vision we have is that the integrins are binding to the extracellular matrix, and in some fascinating way, they recruit lots of intracellular proteins. And we're trying to kind of understand what they do. One thing we know is they link to the actin cytoskeleton. So here, the red is the actin cytoskeleton linking to the ends of the integrins, but we think there's an awful lot of proteins just to do that one function. So again, coming back to this diagram, the complexity is there's really quite a lot going on inside the cell, and we've been trying to understand what all these proteins do and how they contribute to the adhesion function. And when I say a lot, I mean really a lot. So this is a diagram from a study looking at various proteomics approaches to define what's called the integrin adhesome, the set of proteins working with integrins. And you can see it's really quite a big set. And this is one that's conserved between multiple models. So this is where actually using Drosophila is quite helpful. So the genome of Drosophila has not undergone the 2 duplications that happen during vertebrate evolution, which means that for any particular component here, there may be up to 4 copies of that in the human genome and only 1 copy in the fly genome. And also by sort of applying very stringent criterion to the proteins, we've actually been able to narrow this down into a much smaller set to focus in on. So here's just some diagrams of these proteins. The main thing is looking at them and the work that's been done on their function actually doesn't really help you really understand what they do. So all these little various boxes, triangles and rectangles represent conserved protein domains, the main function of which is to stick to things, either other proteins, the membrane, or actin, for example, another protein, sometimes lipids in the membrane. So this one, for example, is called integrin-linked kinase, but it turns out it's a pseudokinase, and its kinase domain is, again, used as a protein interaction domain. The one real active kinase is focal adhesion kinase. So we're kind of left with, well, why do we need all these sticky proteins? How do they make integrin function work? So again, what we do is take the approach of knocking each of those out in Drosophila and seeing what goes wrong. So this again shows the sets of protein, and highlighting over here is just some examples. So here's the integrin that you've seen already, very nicely localized. And when it's bound to the ECM, it then recruits all these other proteins. So I've shown 3 here, but that it's true for everyone in this group. And then when we knock them out, we get a range of different mutant phenotypes, which are sort of shown along here. So starting at the bottom, this actually shows another morphogenic event that fails in integrin development, where it fails to what's called germ band retract. So this is a null integrin phenotype. We get the same phenotype if we knock out these 2 proteins, talin and Fitzkindlins. So that means that they're really essential for integrins to work, because if you eliminate them, you get all the same defects as you get when you eliminate integrins. Now these proteins, which are also recruited, here's one of them here, have a milder version. They still die as embryos. What's shown here is the actin that's retracted within the muscle, so it can't anchor properly. But you see they don't have this germ band retraction phenotype. Then we take this set here, and they all survive till metamorphosis in the pupal stages. They die as pupae. And furthermore, these ones then survive as adults, but they have these blisters phenotype. And you remember I showed you a picture of making a patch of mutant cells for integrins in the wing gives you a blister. That's actually true for all of these mutants here. Clones of those will give you a wing blister phenotype. And then somewhat mysteriously, we had 4 here with no phenotype that we could detect. So that's just in a way means we haven't looked hard enough. We need to push them more to identify what it does, because you would not conserve these proteins, so these are well conserved between Drosophila and humans, if they weren't doing some important function. Okay, so I'm going to start with telling you some of our work about talin. So we know that talin really does an important job with integrins. So how does it do that? So as mentioned, yeah, talin is required for all of integrin's function. So we were kind of thinking it does one really key thing. And what I'll show you is the kind of surprise is that the mechanism, the way talin works, varies in each developmental event that you look at. And that was revealed by finding that different parts of talin were required for different functions. So just a little bit of explanation about talin. It's quite a big protein, and it has at its N-terminus the ability to bind directly to the integrin cytoplasmic tail. And at the C-terminus, it has an actin binding domain. So the current model is that when it engages integrin and actin, then the pulling of actin actually stretches talin and unfolds it. And that then exposes binding sites along talin for a second protein called vinculin, which can be recruited and itself can recruit additional actin and stabilize this structure. So this is really, then talin is really the first, is really one of the best understood examples of how mechanotransduction can work. By pulling on this protein, so mechanical forces, we're unfolding domains with it, and then that permits a chemical change, which is the redistribution of the vinculin molecule. Talin is a mechanosensor or mechanotransducer, and vinculin, one of the best known mechanoeffectors. So we did a genetic screen and isolated a number of mutations across the whole talin molecule. So here's again this head. It has its integrin binding site 1 here, integrin binding site 2 is there, the actin binding site, the C terminus. And we isolated a whole series of truncations, and we also generated one more directed, which deletes this head domain. And so then we explored whether any of these mutants still retain function. So we looked at 3 different developmental processes, so the muscle adhesion and the wing blister phenotype, but also that epidermal morphogenesis process of germ band retraction. And the kind of surprise was that we got different results for each. So, for example, if you delete the head domain, then it's essentially can't function at all in muscle attachment sites, so it's absolutely essential. Whereas germ band retraction occurred normally, and adult wing adhesion, we got a kind of partial phenotype. Similarly, if we delete this actin binding site, that partly eliminated function in the muscle. It was absolutely essential for germ band retraction, and then also partly a function for wing adhesion. And then this integrin binding site 2 does not seem to be essential for the muscle. So if we delete the actin binding domain, we get this partial phenotype. If we delete the IBS2 as well, it doesn't get any worse. Whereas in the wing, it goes from a partial phenotype deleting this to a null phenotype if you delete both of them. And because this is already fully essential when we delete that, we couldn't test whether that was necessary or not. So we kind of put this together in this kind of model where we thought, well, in the muscle, I won't go through the logic that got us there, but this was where we got to. In the muscle, basically, it's in this orientation, pulled away from the membrane, whereas in the wing, we thought it was in a parallel orientation. So we happily sent that paper off, and they came back, and the reviewers said, no, well, you have to prove that they have this different orientation. So we were able to use this technique called FRET. So FRET occurs when you have 2 fluorescent or light-absorbing molecules close to each other. So when this red one is in close proximity to the green, it affects the lifetime of the fluorescence that the green one emits. And you can measure that using a sophisticated confocal microscope that uses a pulse laser. So you can do a pulse and then actually see the timing of the fluorescence emission. So there should be another talin rod here. My PowerPoints were doing weird things on the train. But basically the idea is we put a red fluorescent protein near to integrin binding site 1 and a green fluorescent protein on the end of the integrin. And our prediction would be that we would get FRET here in the wing in this model, but no FRET here because they're not close enough. And that's indeed what we found. So basically, this is so here we've got a pseudo-colored scale which tells you whether there's FRET or not. And the more blue it is, there's low FRET, and then as it gets orangey red, you get FRET. So basically that's showing you that in the muscle attachment sites, we're not detecting FRET, but in the wing we are. So that was good, but then they said, well, maybe it's just a little bit away. It's not as dramatically away in the muscle as we thought. So we should go back and use super resolution microscopy and actually show that they have different orientations. So we went away and did that. So this is, so I don't know if you've yet come across super resolution microscopy. For a long time, there was a sort of biophysics law which argued that the limit of resolution was completely determined by the wavelength of light. So, you know, the wavelength here is about 500 nanometers, and that meant we could not distinguish two dots any closer than 250 nanometers. And your average protein is maybe 10, 20 nanometers, so that's not that good to really see the way proteins interact. And people have figured out a few different clever ways to get beyond, break this resolution one. And this one is one of the easiest to use. It's called basically, what is it? I know the acronym, but it's using really two gratings and a computational method to increase the resolution. And so what's shown here is zooming in on the muscle attachment site. You can have different color schemes, but it's the same data, where the integrin is in green or yellow, and then the tail of integrin is red. And you can see in the muscle, they're clearly resolved, which fits with the two muscles attaching end to end, the ECMs between them, and the talon is being pulled out in each direction. So the C-terminus is pulled away from the integrin. Whereas when you do the same kind of imaging in the wing, we can't resolve the two colors, which fits with them being all in the same plane. Good. So basically, what we're kind of excited about is that these different orientations tell you that maybe it's using it to sense different kind of forces during the morphogenesis of the tissue. So maybe in the muscle, the forces are much more perpendicular, whereas in the wing, perhaps they're more stretching or in the plane. And similar thing in germ band retraction. I think the other really important take-home message is that if you're doing, if you get different results from your competitors, it could be you're using a different cell type and it does work differently, and you both could be right. So don't expect the molecular mechanism to be identical in all situations. And basically, so talon has two roots, a more strenuous upright root in the muscle, and then the more relaxed root in the wing. Okay, and I think this just surprised us that even this what really core component that is essential for integrin, all integrin functions, does it in different ways in different places. So that was sort of trying to take apart the particular molecule was a helpful way to tackle that. And what can we also use by looking a bit at how they arose in evolution? So if I come back to this diagram, just to remind you that we used fruit flies to simplify our set of proteins that we were worried about. So can we go to other animals that are related to us, but more distantly, for example, sponges, and look at them, but in fact, they have the same exact set of adhesion molecules. So we have to go even a bit further to the unicellular ancestors of animals. So you may not be so aware. So this is a tree of eukaryotes. So we're all eukaryotes are derived from one common ancestor. It's called the last eukaryotic common ancestor. And we know that because all eukaryotes have mitochondria or had mitochondria at one point. So that event of acquiring a mitochondria was a very singular event in evolution and permitted all eukaryotic evolution. This is a tree of different eukaryotes that are currently living in Earth, and you can see that they all branch very low down in this tree. So that means there was a lot of initial diversity into these major groups, which are really very different from each other. So I think of myself as being quite different from a plant, but there's 1, 2, 3, 4 other groups that are really just as divergent. So if we ask when did the integrin machinery arise, it turns out it's really limited to this grouping here, including amoebozoa, which branches off very basally here, and then the animals. So we're going to zoom in on some selected individuals in this. Okay, so I'll just spend a little time on this tree. So basically we've got, I've put sponges as the most primitive metazoa at the top here, but it could be Drosophila or human. Down here we've got the universal common ancestor that gave rise to bacteria, etc. So the only thing that we share of this machinery with bacteria and archaea is actin. When you got the first eukaryote divergence, they have a set of proteins here. I haven't really discussed these ones except for PIX, was in our previous one. So these are a few other related proteins. So these are found in all the organisms in this tree here. That's what that indicates by being at this point. And then we have a branch now to, so Naglaria is really in a different branch than the bit I was highlighting with the red triangle. So if we look at this set of proteins, they're present in this interesting organism called Dictyostelium. So this lives as a free-living amoeba, but when it runs out of food, it actually does a little development thing and forms this sporulating body, forms a little larvae that crawls around, then sporulates. So this was kind of a shock for me because all of these proteins in a fly or in a human are really dedicated to helping integrins function. But here they actually are in an organism that doesn't have integrins. They must have be able to do other things, either with other receptors or with other parts of the cell, independent of integrins. So then at this branch, this organism here now has integrins, and gained these two ones, ILK and parvin. And then in this next branch, we gained this protein GIT. And if we look at this organism, this is another slime mold, but distantly related to that one, they actually have all these proteins except they've thrown out the integrins again. So we'll keep all the inside stuff, but chuck the integrins. And then this organism, it's a very simple looking cell, has been a really fascinating one because its genome contains many proteins that we used to think of were required for development. Tyrosine kinases, for example, brachyuria, a key transcription factor required for pattern formation. And of interest for us, it has two of these proteins, this focal adhesion kinase, a tyrosine kinase, and a protein called tensin with adapter domains. And then we've got additional things that are unique to the metazoa. So a few thoughts about this. So one is this suggests that integrins co-opted existing machinery. So this is an interesting idea in evolution. In other words, we can gain a molecule such as integrin, and it grabs things that were already there doing other stuff and says, okay, now I'm in charge of you and I'm going to use you for my purposes. And there are increasing numbers of examples of that kind of process occurring because the things are there first and then you use them later. We think, for example, from our work, which I won't have time to tell you about, but about with fits and kindlins, that one of the things that does is make this whole adhesion machinery stronger so it can withstand the larger forces in multicellular animals as you got bigger cells. And then I'd like to talk a little bit now about this. So it was kind of intriguing that these two proteins appear at the same time. It kind of suggested that it might work together, which at the time when we did this analysis, we had no idea that that was true. There was no evidence linking their function together. So that we then got in an independent project some evidence that that did in fact put them together. So if I come back to this, we were a bit perturbed by these proteins up in this part, which had so little function. So we started trying to look at whether the flies were as athletic as their the ones that lack these proteins were, as robust in their movement and so on. But eventually we started looking at the structure of the muscles. But I wanted to just highlight this point, which is coming back. So how do we interpret these proteins? Why are they not as important? So in a very linear pathway, if you knocked out every part of it, you'd get the same phenotype. And this one is clearly, we've got a set of proteins which clearly have varied function. So one way to interpret that is that it's still fairly linear, but you get redundancy between two things. So if these two proteins can do more or less the same thing, then if you knock out this one's still doing a major job. Or similarly, these guys all might together do the same job. So if you knock out any individual one, nothing really bad happens because the other ones can do it. So that is called redundancy. And one of the ways to tackle that is to then make double mutants or between them and see if the phenotype gets much worse. The other option is it's more of a tree where there's you diversify, each has a very unique function, but it's together, but it's a more minor part, and this has itself function, this has function. So these ones in the ends are contributing a more subtle function at the end. So this is so we started looking actually at the adult muscles, which are interesting because they're structured a bit differently. They're more like our muscles and they're formed of these myofibrils. They're also kind of amazing because when the fly is actually flying, they're contracting at 200 times a second. So they're really, when the flies are flying, they're really under quite rapid contraction. And you can imagine that would pull hard on this integrin junction. So this is looking at, for example, vinculin at the ends of the muscle here. So we thought maybe this would be a more sensitive system to identify the function for some of these other proteins. And this just shows that we could actually identify rather subtle defects, so some stronger than others. This one is slightly bigger than in the wild type in all these different mutants. So that was good. It just showed they weren't perfect, but these proteins are doing something, but you have to look harder to find them. But then when we made double mutants, we saw no sign of redundancy. So we just got the phenotype of adding those two defects together, whereas if they were sharing some other major function, then you would expect it to get much worse. So that kind of nixed that model. So then, but when we did a double mutant with FAC, actually, we got this surprising result that things got better. So it's not completely wild type, but the pulling away that you would see here is much reduced. And we found we could get the same effect with a mutant we have in the integrin, which makes it a bit more active. And this actually fits with something I haven't elaborated on yet, which is that we think integrins can exist in a kind of closed inactive state and can be activated to bind the extracellular matrix. And for example, talin, in addition to linking to the actin cytoskeleton, is thought to contribute to this activation step. And we know that FAC inhibits integrin function from, again, if you knock out FAC in mammalian cells, the integrin adhesions get bigger, they don't get smaller. But we didn't really know how it worked. But so, okay. But basically, our data would support the idea that with either of these mutants, you can increase the activity of integrin and therefore suppress some of these weaker defects. And We could also suppress lack of vinculin by elevating integrin or FAC. I'm not going to show you that, but not tensin. So we could get some partial suppression again with the active integrin, but not with tensin. We still get these muscles pulling away. So that basically indicated to us that that's because FAC, for it to suppress integrin, it needs tensin. So without tensin, the loss of FAC doesn't result in the elevation of activity. So we think tensin is either helping integrins activate or activating itself, and then that is normally inhibited by FAC. So that was nice because it fit with the idea that these two proteins work together. But then we still had this mystery of, well, why do we need this mechanism to regulate FAC? And this just shows you, it's a little bit hard to see, but normally, if you look now by electron microscopy, you see that the integrins are in these discrete plates, which fits with the angle of these protrusions. And what surprised us is in the FAC mutant, those were continuous. So we think that FAC is there to modulate which parts of the membrane integrins are active in. Okay, so with this kind of thing, we're beginning to sort of pull apart the different components and get a sense that they have different roles in this thing. So I've talked a little bit about this core connection and mechanotransduction of talin. We think that this complex may help stiffen the cortical cytoskeleton. This one is more involved in signaling, so it's required, for example, to down-regulate filopodia. And then this one is sort of regulating the size of the adhesion plaque. And we kind of like to think about this like cocktails. So you have these key elements which are all important to make a good cocktail, but the variation in amounts gives you a range of effects and functions. And so this is our kind of vision of these different components really together, working together in a joint way to mediate integrin function. So basically, we think that the reason we need so many is it's about the diversity of the way integrins work in different contexts. And I think if coming at it from a sort of cell biology and developmental biology context, it really, really helps to be looking at things within an intact organism, which does a lot of different kinds of things. So if you're studying it in just a single cell in culture, you have a very limited number of assays you can do for its function. Whereas if you're looking in an intact organism, so every time we look in a different tissue and study what integrins do, we learn something new because it works a bit differently. And so we're beginning to understand some of these subfunctions, but we're still plenty of place to go to really understand how these components contribute to integrin function. And we're trying to take more biophysical approaches to really look at the molecules live at the moment. And just finally, I wanted to highlight two people, Benj Klappholtz in my lab, who really did the work on talin, and then Hannah Green, who did the work on tensin and FAC in the adult muscles. And that's it. Thank you for your attention.